Hello, my name is Patrik Bohman and I work as an environmental assessment analyst at the Swedish University of Agricultural Sciences in Sweden. I'm going to talk to you about a common benchmark study on crayfish and crayfish plague. But first of all, why do we need these kinds of benchmark studies? Well, crayfish species are quite hard to detect from water samples. There are no standards yet, so a lot of different methods and protocols are used to sample eDNA and detect crayfish and crayfish plague. Therefore, we need to focus on a common methodology for these particular species. We also need to evaluate which practical methodology that works best. We conclude that benchmarking is a good way to make comparisons to see how robust and precise the different methods and protocols really are. Benchmarking also allows us to define better suited quality standards and practical guidelines to detect crayfish and crayfish plague in water samples. Now let me give you some background. In 2020, we held a workshop on the most used methods to sample and analyze eDNA from crayfish and crayfish plague. 34 participants from 8 different European countries attended. The workshop was about how to detect crayfish and crayfish plague from water samples. And the emphasis was on practical issues like sampling techniques with direct filtration, the use of filters, error handling in the field, transportation, preservatives, qPCR, etc. The conclusions from the workshop were that we need to move forward to compare our different methods in an experiment. This experiment will start in 2021 to 2022. And in the experiment, we will use different aquariums. These aquariums will contain different biomass, that is, numbers of crayfish. And we will also include differences in water quality to induce different grades of inhibition to the samples. Participants will then use their own equipments, methods and protocols to sample water, preserve the filters, extract the DNA and analyze with qPCR or ddPCR. Finally, we will compare the results and come forward with improvements and guidelines. Signal crayfish will also be screened for prevalence of crayfish plague. Thank you. And if you are interested to join this benchmark study, please contact me, Patrik Bohman. Thanks.